All right, so this morning I'm going to talk about anemia, infection and iron metabolism with a focus on the tropics, so low middle income countries where there's a high exposure to infectious diseases, and we're going to focus a bit on malaria as a specific case. So I'm going to start with just talking about what are the causes and effects of anemia, particularly in these tropical settings, then lead into a bit about iron metabolism, how to assess iron status and the problems of assessing iron status. And then we'll talk about iron supplementation, and this is where we talk about it as a potential double-edged sword, because there can be issues with iron and infection. And then I want to introduce you to a new molecule called Hepcidin, which we hoped would help us understand and solve lots of these problems, but so far not quite yet. So just to touch on again that anemia, so anemia means basically a lack of blood, so insufficient blood, or red cells to be more precise. So the red cells carry hemoglobin, which is responsible for carrying oxygen to the tissues. So here we look at the global prevalence, and although this data is now from 2008, it hasn't changed a whole lot. The global prevalence might have decreased a little bit in some of these countries, but not much. So here you see the prevalence by the Human Development Index. So this is the highest income or highest HDI countries and the lowest, and these are the different population groups. So here we've got preschool age children, pregnant women, non-pregnant women, school age children, men, elderly, everybody. So you can see that even in these high income countries, anemia is still somewhat prevalent. So in preschool age children, over 10%. This would be mostly mild anemia. So although the problem is much, much greater in these lower middle income countries, and particularly in countries in the tropics, it is a global problem that all countries have concern about. But you can see the groups that are most at risk are the ones that are often the only ones that are talked about. So preschool age children, you can see 66% deficiency here in this group. Pregnant women are next, and then non-pregnant women. But even in men, and men often get forgotten about in anemia. No one ever talks about anemia in men, but even in men, you're 21% are anemic, and that's going to have, again, important public health consequences. And the elderly are another group at risk. So just for your reference, here are the WHO standard cutoffs. It gets a bit confusing because there are some different cutoffs that are used by different groups and different sources, but I think it's best just to stick to these ones. But this is just for any anemia. So there's different cutoffs for mild, moderate and severe, and again they may vary depending on what reference you're looking at. There's also now a new reference for children 0 to 6 months, but that's a particularly difficult group because their hemoglobin changes as a result of other factors that I won't go into, but particularly in the first few weeks and month of life. You're also, in the past and clinically, people often talk about the hematocrit, which is when you spin the blood sample and then you look at the height of the red cells that should sink to the bottom as a proportion of the height of the sample. So it's very primitive. You can also get it as an output when you run an automated blood analyzer. And people used to use it, but really if you've got a hemoglobin, there's really no point in looking at hematocrit unless you have some other specific reasons. And the hematocrits often don't correspond very well with these hemoglobin cutoffs, and because it can depend on how you process the sample, how hard you spin it, how long you leave it before you read it. I don't recommend using hematocrit. So as I said, anemia is from an insufficient or not enough red blood cells, and that can be either because not enough are being produced or because too many are being destroyed. So two main reasons for being anemic. And then this is the reference that this table comes from, and this remains probably one of the most useful references for people like yourselves. It's from 2001, but the basics haven't really changed. So why do we care about anemia? Because anemia can decrease physical work capacity. Because if you remember that the red blood cells are required to transport oxygen to the tissues, and obviously you need oxygen to metabolize energy to do physical work. So your muscles need oxygen to metabolize. So that's why anemia in men is potentially also important. In populations where the men are still doing physical work, then even being mildly anemic is likely to reduce their physical work capacity. And there were some interesting trials done in the 70s, which I haven't really shown here, but where they looked at supplementing, one was tea plantation workers and one was rubber plantation workers. And I think it was in the tea plantation workers one, where they showed that they got paid for the amount of tea they picked. So the group that got iron supplementation picked more tea and got more money. And in the other study, the supplemented group didn't do more work, but when they looked at how they spent their energy and what they did, they then spent more time on non-work activities. So when they got home, instead of just going, they had time to do more jobs around the house and to play with the children, for example. So other important effects. So anemia is also associated with increased risk of infections, and we'll talk more about that, but probably because anemia has multiple causes. And then we know that anemia is associated with reduced cognitive development, and that iron is required for cognitive development in children. And therefore also anemia is associated with growth and also lifelong earning capacity. And then it's very important in pregnant women because increased risk of adverse birth outcomes, premature births, and increased risk of neonatal death, low birth weight, and importantly, those children are going to be born with low iron stores. So if the mother is iron deficient, she will put as much iron as she can into the infant at her own iron stores, but at some stage the infant might not have enough iron because a newborn baby should be born with enough iron stores to last them the first 6 months. And that's because breast milk only has a very small amount of iron, and therefore that has to supply their iron need until complementary foods are introduced. So here was an analysis showing the global burden of years lived with disability due to anemia. And perhaps unsurprisingly, you can see that the problem is greatest in sub-Saharan Africa and highest in these countries here, but also Asia. And of course, Asia has South Asia has such a large population that that's a huge number of people with anemia. So to summarise, effects of iron deficiency. So children, cognitive impairment. And one of the issues is that A child's brain is developing up until sort of 2-3 years of age. The actual structure and function of the brain is being laid down at that time. So if a child is born iron deficient and is iron deficient before sort of 2 years of age, it's quite likely that supplementing iron after that time is too late. The damage is already done. So we know that iron is required for forming the myelin sheath on the outside of nerves. And you need that, which is the white matter that you see on the brain. And we need that for insulating the nerves so that the nerve conduction is efficient. So that's one of the problems, is trying to prevent iron deficiency in young children in order to reduce cognitive impairment. However, you should be aware that even in adults, there is some evidence that supplementing iron in iron-deficient adults can improve certain cognitive functions, such as memory. And also, the other thing to remember is that some of the effects we see might be due to the anemia as well as the iron deficiency. So anemia itself is about, A, there are other causes of anemia, and B, if you don't have enough red blood cells to deliver oxygen efficiently, some of the effects may be due to the anemia itself. So pregnant women, potentially they have a reduced iron supply to the fetus and low birth weight babies. And we know that low birth weight babies have an increased risk of long-term health adverse outcomes, so including mortality and also later health issues in adult life. And then adults, effects on anemia, reduced work capacity, and then poverty. So the stages of iron deficiency. So iron is stored in the liver in quite large amounts, and it's stored in a protein called ferritin. And when we measure serum ferritin as a marker of iron status, what we're attempting to measure is a reflection of how much iron is stored in the liver. Because in a healthy person, the amount of ferritin circulating is proportional to the amount of ferritin stored in the liver, which contains iron. So the first stage of deficiency is your stores going down. And then later on, once your stores are really running out, then you get decreased supply to tissue, which might be shown by other iron markers. And then the final stage is anemia, when there's insufficient iron for hemoglobin synthesis. So iron is a requirement for the heme component within hemoglobin. And it's important to remember that at this stage there could be functional effects. So because loads of tissues require iron. So at this point is where you might be getting effects on cognitive development, for example, in young children. So before they're anemic, you might have iron deficiency that's having consequences. So where do we get our iron from? So iron, there's basically two main sources in the diet. So there's heme iron, which is from animal sources, which is basically from the heme within hemoglobin or myoglobin, which is also in muscle tissue, from those animal foods. So iron from these sources is generally very well absorbed. But there's also a lot of iron in plant-based foods, and it's perfectly possible to get enough iron from a vegetarian diet as long as your diet is varied, and it depends a bit on the food matrix. So there can be various inhibitors for non-heme iron. So an important one potentially is tannins in teas and also in some other drinks. So for example, drinking tea with your meal is perhaps not the best idea, because it will be inhibiting the iron absorption from that meal if you're drinking tea at the same time. And then things like phytates that are very high in some staple foods, depending on from different flours, and also in things like unleavened bread can be high in phytates. So like flatbreads are much higher in phytates than breads using yeast that have kind of gone through a different process that reduces some of this phytates. And then vitamin C helps to promote absorption of non-heme iron. So again, a nice varied meal that contains fruits and vegetables will have improved absorption because of the vitamin C in it. And then I've written sort of general, here it's acting as a reducing agent, but actually a little bit of meat, because there's certain amino acids that are more prevalent in meat, actually will also enhance the bioavailability of the non-heme iron. So again, this is why young children who have the high iron needs and their weaning foods are often very low in bioavailable iron, they're the ones that need a little bit. They don't need a huge amount, but even just giving a little bit of meat will also help to absorb the non-heme iron. So we said these weaning foods, which are often like the staple foods, so provided without meat or without the vegetables, just the staple, have insufficient iron. And in high-income countries, a lot of these weaning foods that people might buy are fortified with iron. And then the other thing that happens with iron is that inflammation and infections cause a reduced absorption and also utilization of that iron that is within the body. And we'll talk about why that is in a bit. So the other thing that's really important to remember, and that many people forget, is that most of our iron needs are actually met from recycling the existing body iron. The amount of iron that we need from our diet is actually very small. So what normally happens is that you've got this big store in your liver, so you've got something like 1,000 milligrams stored in your liver. You've also got, let's say, another 400 or so milligrams in other tissues. And then the only way that you lose iron normally is a very small amount that's mostly lost through losing the cells lining your gut. So every day, just as a normal process, these cells are being lost. And they contain iron and zinc. And so normally, your iron loss is very small, 1 to 2 milligrams a day, just from losing cells. And obviously, reproductive age women, and when they're menstruating, that's why they have higher iron requirements, because they are losing iron every month from blood loss. So in a normal, healthy situation, the amount of iron that's absorbed from the food will match the amount that's lost. And that's only normally around 1 to 2 milligrams a day. Whereas the amount of iron that you need to make new red cells every day is much, much bigger. So you're turning over 20 to 25 milligrams of iron a day, making new red blood cells in the bone marrow, which is where they're made, because the lifespan of your red cells is around 90 to 120 days. So you're making new red cells every day to replace those cells. So here you've got iron stored as ferritin in the liver, and then iron is transported on something called transferrin, and it's transported to wherever it's needed as transferrin. So it's going to the bone marrow, making blood cells, and then these red cells, when they age, they are recycled. So under normal circumstances, these macrophages, reticular endothelial macrophages, can detect old or damaged red cells, and they gobble them up. And then they digest the red cell, and then they put the iron, they take the iron basically back to the bone marrow to be reused. So at any one time, these macrophages also contain a large amount of iron, which they've recycled from dead or old red cells. And most of the time, these macrophages will recognize these old and damaged red cells before they actually die and break apart. But there are other mechanisms for scavenging hemoglobin that's released from dead or lysed red cells, which also feed the iron back into the bone marrow or feed them into these cells. So the way we control our iron status is through absorption. We do not control how much is lost. So we regulate how much iron is absorbed. And this amount happens regardless. We're not able to control iron excretion. So iron is lost normally only through the gut cells, through blood loss, and occasionally during very high red cell destruction, which overcomes this scavenging capacity to recycle the iron from the free hemoglobin that's released. So in most conditions, it's all recycled. But if you have very severe malaria, it can be known as Blackwater Fever. It doesn't happen very often. But it was seen in Europeans, adults, who on first malaria can get extreme symptoms. And there's so much hemolysis that the scavenging can't keep up. And the urine becomes black because of all the free heme that's released into the urine. So under normal malaria conditions, this is not normally happening. It's only if it's very acute. So if we think back to those stages of iron deficiency, so I said that you have iron deficiency in its sort of medium stages doesn't result in anemia. It's only when it's more severe that it impacts anemia. So you've got iron deficiency here and iron deficiency anemia. But there's also all these other causes of anemia. And nutritionists in particular often forget about these causes. And when they talk about anemia, they're often really talking about iron deficiency anemia, but they just say anemia, which is incorrect because there's all these other causes. So can some of you suggest what some of these things here might be? Without looking at your handout.
Cancer, for example.
Cancer, yes. Can you explain a bit more why?
Something kids.
It's on.
Yeah, I'm not very sure how, but I guess some in case of cancers, we may have the destructions of the bone marrow.
Yeah. Okay. Yeah.
That's the most obvious that's coming to me now.
Yeah, which is probably from the treatment of the cancer,
Right.
which might be targeting the bone marrow. Yes. I might come back to another reason. Let's see what other people say. Anyone else?
Hidden bleeding in gastric.
Yes. So bleeding, although that's probably going to impact.
Or, for example, parasitic problems.
Yes, parasites that affect blood loss mostly.
Okay.
Yeah, hookworm. Hookworm's the classic example. Any others? Yeah?
Pernicious anemia, sickle-cell anemia.
Okay, yes. So other causes of anemia, pernicious anemia, which is B12 deficiency occurring in the elderly because of the inability to absorb it because of antibodies to intrinsic factor, which you need to absorb it. So that's B12. So there's another micronutrient that's needed to make red blood cells, the B12. And then you also mentioned sickle cell disease. So sickle cell disease causes anemia how? Very simply.
I think the shape of the red blood cell is altered, and so the cells are destroyed.
Yep, that's basically
I
it.
think it loses the flexibility to go through small capillaries due to the structure of the membrane
Yeah.
that is altered.
Yeah.
But I can't remember which structure.
So sickle cell disease is a genetic disorder, which I could have given you a whole long lecture about sickle cell, which I would have liked to have done, but no time. So sickle cell is a genetic disorder affecting the red cells so that some of them under low oxygen conditions form this sickle shape. So then they don't function as well, and their lifespan is much, much shorter. So the lifespan of red blood cells with somebody with homozygous sickle cell disease might be as short as 10 days. So normally it's 90 to 120 days. So they're being destroyed so fast, the bone marrow just can't keep up, hence anemia. So it's about that balance between destruction and generating new ones. So in extreme hemolysis, like in sickle cell, or extreme destruction of red cells, the bone marrow can't keep up. So we had another micronutrient deficiency. What about any other micronutrients?
Folic acid.
Yes, okay, folic acid. You need folic acid because when you make red cells, or when you make any new cell, you need DNA, and you need folic acid to make DNA. And in the early stages of making red cells, they have DNA. So, because your first thought might be, but red cells don't have DNA. But they do in their first early stages, and then they lose it. And the demand for DNA from making new red cells is huge because you make so many every day. So one of the early symptoms of folic acid deficiency is anemia. So we've got B12, folic acid, anything else? What other micronutrients?
Vitamin C, I have heard that helps the iron to be absorbed.
Yeah, so vitamin C will help the iron to be absorbed. Doesn't directly cause anemia, but yes. What else? There's two other key micronutrients that we've talked about that also can be important.
Proteins.
Yes, we don't. Micronutrients. What other micronutrients have we talked about?
A random guess, I would say zinc.
Yeah, zinc. Zinc is less clear, but it probably is, maybe in a slightly roundabout way. And then what's another one we've talked about? A really important public health micronutrient that supplementation reduces child mortality. Okay, I might go home now because I'll give up. What micronutrient supplementation to?
Vitamin A.
Thank you. Right, I might be about to resign as a teacher. So vitamin A, again, vitamin A is probably important in anemia as well. And that's because also vitamin A affects the way zinc is metabolized. Zinc also affects vitamin A. Vitamin A can affect the way iron is metabolized. So you have these multiple micronutrients. All right.
You also talked of malaria.
And malaria. And again, how does malaria cause anemia?
Due to increased destruction of red blood cells, which is not matched by production.
Yeah, basically. So malaria causes anemia by destruction of red cells faster than it can be kept up. Does malaria directly cause iron deficiency or iron loss? Normally.
Iron loss.
Maybe at the recycling the iron, there is not enough iron in the blood after bleeding, maybe.
Well, so malaria doesn't make you bleed. And malaria, unless it's very severe, doesn't make you lose
The
red cells,
destruction of this-
but they're recycled. So remember, unless you are losing red cells in the urine, which you do in Blackwater fever, or they're being broken and you're losing the heme, which is the bit that contains iron, into the urine, which is only extreme infections, you're not losing iron. It's still there, and it's still available to be recycled. So this is something that I really want to make sure that people understand, because it's something that's misunderstood by many people. So malaria, in most cases, does not directly cause iron loss or iron deficiency. It causes red cell destruction faster than they can be made again. But if somebody, once you've treated the malaria and you've got rid of the parasites, that iron is still there. So then you can make the new red cells and the hemoglobin should recover. So in a normal acute episode of malaria that lasts acutely for maybe, let's say, a week, and then it's treated effectively, and you have a good diet, and you haven't got lots of other causes of anemia and iron deficiency, your bone marrow will recover using the iron that was sequestered from all those destroyed red cells. So that's an important thing to remember. Let me go on to... Okay. So we've said pretty much all of these things. So contributing to iron deficiency is that diets that are low in bioavailable iron, so hence weaning age children often have the highest prevalence of iron deficiency because the foods that they're given don't contain enough iron, and there's not iron in breast milk. And then, of course, there are high requirements in children and, that should say, pregnant women. Pregnant women because they need to supply iron to the fetus. Reproductive age women because they are losing blood through menstruation. And then other causes of blood loss could be hookworm. Also schistosomiasis, depending on the kind, can cause blood loss in the urine. Some other infections, kidney infections, can cause blood loss. But also trauma. Again, if you have an accident and trauma, then you're going to lose lots of blood. And then anemia, we've got our other micronutrients. And then again, a diet that's poor in bioavailable iron is highly likely to be low in just about all micronutrients. And folate is found mostly in green leafy vegetables, which can also be a source of iron. So a diet that's low in bioavailable iron is likely to be low in other micronutrients as well. And then you've got your genetic causes. For some reason, sickle-cell disease seems to have been lost from this one, but it should say sickle-cell disease. And then there are other genetic diseases that affect either red cell production or red cell destruction. So something called glucose-6-phosphate dehydrogenase deficiency. And the thalassemias also can be relevant. And then we've got malaria because it causes destruction of red cells. And then, but also, general infections and inflammation can cause what we call anemia of chronic disease, or anemia of inflammation, or inflammation-associated anemia. And that's because what happens during inflammation and infection is that iron is sequestered away. And the idea is to keep it away from potential pathogens. And so the production of new red cells is often suppressed, and the iron is sequestered in those reticular endothelial cells. So it's there, it's just that it's not being used, and that can result in this anemia of chronic disease, which is what you would get in cancer as well, potentially, because you've got this sort of inflammation going on. But obviously anything that damages the bone marrow is also going to cause anemia, because that's where the red cells are made, and white cells.
In cancer, some medicines like interferons, they can lower the blood, but it's not iron.
Yeah, they're affecting red cell production, or the inflammation is affecting red cell iron use.
I don't know the word,
Lysis?
Lysis, they make lysis.
Okay, so some drugs might directly cause red cell destruction by lysis of the cell. Yeah. So just to reiterate, so anemia can be from impaired red cell production, which the most common cause is going to be nutritional. But you can also have anemia of chronic disease, where the inflammation and acute phase is preventing that production of red cells. But at the same time, inflammation and infection also reduces iron absorption. So if you have a malaria episode of a week and it's treated effectively, then as soon as you're well again and the inflammation has gone, then you will start to recycle that iron effectively, and you will absorb iron if you need it. But if you have a sort of constant low-grade chronic infection, then eventually you might become iron deficient as well, because your absorption is impaired. So this is why malaria doesn't directly cause iron deficiency, because it doesn't cause you to lose iron. But if it goes on, like any infection that's sort of chronic, it will reduce your ability to absorb iron and use the iron that you have. So we've talked about these genetic defects in hemoglobin synthesis, also kidney failure, because kidney produces erythropoietin, which is a hormone that stimulates the bone marrow to make red cells. And then things that cause red cell destruction, malaria and these genetic disorders, and also there's a viral infection called parvovirus that could cause a very sudden acute drop in red cells from destruction. And blood loss. So again, hookworm, trauma, surgery, etc. And also increasingly we're recognizing that it's not just hookworm, but also some other helminths which weren't traditionally thought to really cause significant blood loss, perhaps cause more than we thought. Okay, so iron metabolism. So we said that iron circulates bound to transferrin. So it's stored as ferritin, and it's transported as transferrin. And then somebody was asking me about this the other day. Transferrin receptors are expressed on any cell that needs iron, so that they will bind any transferrin that's going past, and then take in the transferrin and take off the iron that they need, and then eject the empty transferrin back out. Well, it won't be quite empty, but they take off some of the iron and then send the transferrin back out. So we can also use transferrin receptors as a marker of iron status, potentially, because the amount circulating reflects the demand for iron by cells. And if there's not so much iron around, they express more and more transferrin receptors to try and grab whatever transferrin is available. So as iron availability goes down, the transferrin receptors go up. We have another slide for it. But of course, if you're experiencing a big demand for iron, so let's say you're recovering from malaria, and so now your red cell system is like, oof, right, we've got to make lots of red cells to recover, then their demand has gone up, so the soluble transferrin receptor will go up, which is not to do with limited iron, it's just that they need a lot more. So again, potential problems with how you interpret this marker. And the problem with ferritin, again, is that here in this case, normally the serum levels under normal conditions should reflect the amount in the liver, the stores. But during infection or inflammation, ferritin is an acute phase protein. And it's a positive acute phase protein, so it goes up. And the reason that we think that this happens, and it's a very important physiological function, is that we don't want any free iron floating around at the best of times. But if you've got an infection, you want to make sure that none of that iron is available for that pathogen. So therefore, you increase the amount of ferritin so that each ferritin molecule is only holding on to, the numbers are not correct, but let's say each ferritin molecule is only holding on to 2 or 3 iron atoms. And therefore, if it's only holding on to 2 or 3, it's holding on very tightly. If it's holding on to 20 or 30, it's much easier for a pathogen to come along and pull 1 or 2 off it. And that goes back to some basic chemistry. But, so that is why ferritin increases, so that there's more ferritin protein, so that it can hold on, each one holds onto a small amount of iron and holds it very tightly, keeping it away from any pathogens. And then, I don't normally show this slide again, but I thought to go back to, this was me as a biochemist, that I love this kind of diagram, and to me it all makes perfect sense, and everyone else is like, But I thought it might make you laugh. So you don't need to learn all of this, but it is kind of useful to show you here. So this is a cell across the gut. Okay, so we've got your heme iron and your non-heme iron. So your heme iron is the iron within the heme that's in hemoglobin, and it has a totally separate route of crossing the gut. And this one is very efficient. So your non-heme iron, which is your iron from plant sources, has a much more complicated route of absorption. And here, what means that your iron, which is Fe, is in its 3+ state, which means it's lost 3 electrons. Again, a bit of chemistry. But for it to be absorbed across here, it has to be in the 2+ state, and it has to bind to this protein. And then it has to go inside, bound to this protein, and then it has to be bound then to this transferrin-like protein. And then it goes up to here, and then it needs to be exported across the gut cell through this protein here. And then it has to go back from 2+ to 3+ and get loaded onto its transporter, transferrin. And as I said, we control our iron status by controlling iron absorption. And most of that control is through controlling this route here, but particularly this bit here. It controls how much we export out of the gut cells and load onto transferrin to then be transported to where it's needed. So the important molecules for you to remember are ferritin, iron store, and transferrin, iron transporter. So measuring iron status. So as I've said, you can measure serum or plasma ferritin. Normally it's serum, actually, ferritin. But the problem is that if anyone has inflammation, the ferritin goes up. And now it doesn't correspond to iron stores. So in the children, often you most want to measure iron status are the ones that you can't measure it in by ferritin, because the ferritin is not reflecting iron stores. There have been suggestions that you can take a higher cutoff. So normally it would be either 10 or 12 or 15, depending on which reference you use. Less than would indicate iron deficiency. Some people will say that if it's less than this in children that have inflammation, you can say that that is iron deficiency. But the problem is that there'll still be many children who might have ferritin way over this and still actually have iron deficiency. So again, on a population basis, using this might be helpful, but in an individual child, it's not really very helpful. Plasma transferrin saturation. So again, transferrin is the transporter, and it can carry different amounts of iron. So under normal conditions, we say that its saturation is normally around 20%. So you could force on a whole load of iron, but its normal saturation is around 20-25%. If the less iron is available, then that saturation is going to go down. The problem is, in inflammation and infection, you want to keep iron away from pathogens, so you're also going to increase your amount of transferrin protein to ensure that it's not too saturated. So again, pathogens can't pull off the iron from it. So then this will decrease in infection as well as in iron deficiency. Then this marker was being advocated for a while as being a really useful, potentially really useful marker. What it is that when you're making hemoglobin, and you need to add in iron into the heme, if not enough iron is available, the body adds in a zinc instead, because otherwise the whole production would just stop. So it adds in a zinc to allow the process to keep going. And then what you get is that within red cells, you get a certain amount of this, what we call zinc protoporphyrin, instead of the finished heme ring. So when there's limited iron, the amount of this zinc protoporphyrin within the red cell goes up. And there's a nice machine that can measure this from a finger prick sample, and you just put it on a little glass slip, you stick it in the machine, and about 15 seconds later, it tells you the zinc protoporphyrin per mole of heme. And you can do that with whole blood. And in theory, this is not directly affected by inflammation because it's not an acute phase protein. So it is useful, but it still won't tell you the difference between this kind of true iron deficiency and this functional iron deficiency. So again, in inflammation and infection, where you have this anemia of chronic disease, the iron might be there, but it's just not being used. So you will still see this go up. And in that case, giving iron might not help because the body's just not able to use it because it's saying, no, we're just going to keep it sequestered. So it is potentially useful, But the other thing to be aware of is that there can be interference in the whole blood from things like bilirubin. So when I tried it in sickle cell, it just didn't work at all because there's too many other things going on. There's interference and it gives you these false readings. And then you can wash the red cells. So you're just measuring the red cells, but by the time you do that, it's gone from being a quick, fast, easy test to something that's really quite difficult. And then you've got this soluble transferrin receptor that we just talked about. So the receptor that's expressed on cells trying to grab transferrin. So it will go up in iron deficiency. But at the moment, there's no standard cutoff because it depends what kit you use to measure it. And also, it will go up in an increased iron demand. So when erythropoiesis goes up, it'll also go up independent of iron status. So it will go up in sickle cell disease because the bone marrow is doing this, trying to keep up. So the soluble transferrin receptor is going to be high, as it will be high after a malaria episode where your bone marrow is increasing production of red cells. Oops. Where are we? Okay, I need to go faster. Right, then another measurement that was proposed is if you take the soluble transferrin receptor and you take the ratio of that to the log of ferritin. So you do a natural log of the ferritin concentration and you use the ratio. And so there was a good study done in Malawi, which showed that if you use this ratio, it's not affected by inflammation or malaria infection. But it was only really validated in severe anemia. So how well this works in more mild iron deficiency and anemia is not entirely clear. And it's not being used as much as I thought it would be. And I'm not quite clear why more people are not using this, apart from expense, because soluble transferrin is relatively difficult to measure and relatively expensive, whereas ferritin at least will run on all your standard clinical chemistry machines that a hospital lab would have, whereas your soluble transferrin receptor doesn't, and you have to do it with ELISA kits yourself, with a person, with a pipette doing this, and you can't just run one test. You've got to buy a kit that does a minimum of 26 or kit tests or something. If you use an automated red cell analyzer or blood cell analyzer, you also get these measurements, which is mean cell volume of the red cell. So iron deficiency, the red cells become small because they haven't got as much hemoglobin in them. But other things can cause red cells to be small. And then you've also got mean cell hemoglobin concentration from your automated analyzer. And in Kamija Firi's study, this one was the next best predictor after this. But again, there can be other factors, including genetic things that affect this. And then there are some machines that are being made particularly from a Japanese company called Sysmex, who I'm working with at the moment. And their machines they measure the amount of hemoglobin in reticulocytes, which are the first early red cells to be produced. So they're not proper red cells yet. And normally, just a few are released. And if you look at the amount of hemoglobin within those reticulocytes, if you look at one point in time, it doesn't necessarily tell you anything, but you can look at the response. So if you're giving iron supplements and you want to see an impact on hemoglobin levels, you've got to wait at least a month before you're going to see a significant impact on hemoglobin, because it takes time to make all those new red cells and for it to go up enough. And that's often one of the problems, is people give iron, but then don't wait to see, or they don't get people to come back to see whether it's actually had an impact on hemoglobin. Whereas this, you will see a response very quickly, because these first reticulocytes are produced within a couple of days. So if the iron is going to be used, you will see the reticulocyte hemoglobin go up even by about day 3 and definitely by day 7. So if you know that the person is taking the iron that you're giving them, and they've taken it for 7 days, and the reticulocyte hemoglobin has not gone up, then you know that they're not absorbing that iron. And so either they're in a state that is not allowing them to absorb it or use it, or they're not iron deficient. There's some other reason for their anemia. Or in most cases is that they're not actually taking it. They might say they're taking it, but they're not. So iron. So we do know that iron deficiency is probably the most common nutritional deficiency globally. And also that iron supplements are the most common medical prescription, but actually treatment efficacy in resolving anemia is actually quite low. And in part, that's because there are side effects of taking them. People don't like taking them. They can make you constipated. They can make your poo look black, which is a bit disturbing. And people were like, ooh, especially a sort of rural population who are not very educated perhaps. And if you don't tell them that this is going to happen, then they're going to be very suspicious. And you can actually get sort of stomach pains from the effect on constipation, etc. Plus, what we're going to talk a bit more about now is that they potentially have toxic effects and increases the risk of infections. So there's been evidence that iron supplements can increase infections and poor outcomes in infections. I haven't got all the slides up, but there's some evidence going back over 1,000 years when it was first documented. And certainly early on in TB, it was recognised that giving iron supplements to TB patients when there were no drugs available was generally a bad idea, and they died very quickly from very extreme sudden increase in TB. But it's been very much forgotten about because iron deficiency is a problem it's common, and we know that it has these impacts on cognitive development of children, so that all the public health nutritionists were kind of ignoring that side of things. We didn't really want to know about it because we've got to get iron in, because we've got to reduce anemia and we've got to reduce iron deficiency. And then there was this trial that was published in, now I forget, I think 2005. And this was a very large, well-conducted trial on Pemba Island, which is an island which is part of the Zanzibar semi-autonomous bit of Tanzania. And so basically the whole island was randomized, all children were randomized on this island to either iron and folic acid, so Phe-Fol, we often say Phe-Fol, so F-E-Fol, or Phe-Fol and zinc, or zinc alone, or placebo. And it was daily supplementation. And then the outcome, and then this trial was actually stopped early by the Data Safety Monitoring Board because there was an indication that the children who were receiving iron in the Phe-Fol only and Phe-Fol zinc groups actually had a 12% higher rate of serious adverse events, which included hospitalization and all-cause hospitalizations and mortality. So,
Mortality for iron supplementation?
yes, there was. Don't forget the whole. All children on this island, I forget the exact number now, were randomized. So there was increased mortality in children who were on the iron groups and increased rates of hospitalization. So 12%. So if you added mortality and hospitalization into one adverse outcome. So here you can see that the blue line is iron folic acid, and then this is the control, and then this one is the iron folic acid and zinc, and then this is hospitalizations. So the cumulative hazard probability for iron and folic acid is higher than the other groups.
Did they research about the reason of this hospitalization?
So most of these hospitalizations were thought to be due to malaria, but there wasn't ideal diagnosis. So you're going from hospital records. So the things that should be said about this setting at the time, so malaria was very common. It was when SP drugs were still being used, so I forget what they're called, sulfur pyridox. Thank you. Say it for me, I've forgotten.
Citrodoxin pyrimethamine.
Thank you. So I call it SP for short. So SP, and there's lots of resistance building at this time. So now first-line antimalarials are artemisinin combination treatment. So that wasn't being used. There weren't widespread antimalarial programs, so bed nets weren't being used. So there's very high malaria incidence at this time, and not very good drugs in use for malaria. So most of the mortality in this setting and hospitalization was thought to be, and probably was, due to malaria. So this caused a lot of shock, and there was a big WHO meeting and review, and it's a little bit out of date now, but myself and my then boss, Andrew Prentice, wrote a very detailed analysis or a review of the potential mechanisms by which this may have occurred. But this really meant that we had to think about how the safety and efficacy of blanket iron supplementation, particularly in areas with high rates of infection. And then soon after this study, there was another large trial which was designed with a very similar design in Nepal, in the lowlands of Nepal, where malaria is very low, but diarrhea as a cause of hospitalization and death was high. And that study didn't find the increased adverse effect of iron. That study was stopped early because there was no effect in the iron group, and they shifted everyone into the zinc group. So both trials were stopped early, and they shifted everyone into the zinc groups, but for different reasons. So that result also made people think that it was only relevant to malaria. So there was something about iron and malaria particularly. However, since then, we think that's perhaps not true, because there's been other studies and other older evidence that suggests that iron is important for a whole range of infections. And the reason for malaria is actually less clear than for many bacterial infections, where we know that the bacterial growth rate is dependent on how much iron they can get hold of. Whereas the malaria parasite, it lives in the red cell. It has access to plenty of iron. It's still not really known how the malaria parasite actually metabolizes and gets iron, but a direct effect from limiting or too much iron is perhaps oversimplification. However, in this study, they did do a sub-study. So in some children had iron status measured, and they used zinc protoporphyrin. And in their sub-study, which wasn't powered to look at this, but it suggested that the children who were iron sufficient, so had enough iron at baseline, were the ones that had the increased risk, whereas the children who were iron deficient benefited. So again, that meant that the suggestions that were coming out was that this is important for malaria, but perhaps not other infections, and it's important to know the status of children. so that you don't supplement children who are iron sufficient. So then the public health implications were that you can't just do blanket supplementation. You've got to measure the status and screen everybody before deciding to give them iron or not give iron. But now that makes the cost and practicality so much greater. So it has huge, huge implications. So we've kind of touched on this already. So I think I've basically said all this. And to emphasize, so iron is essential and often limiting for all organisms. So this is a diagram that Andrew and I developed that we think summarizes the situation between the host, which is us, and pathogens. So there's probably a reason why iron deficiency and iron deficiency anemia is one of the most common deficiency disorders, because throughout our evolutionary history, we've probably been in what we call a battle for iron between us and pathogens. So there's probably a sort of happy medium place, which is a compromise. So there's probably a balance between the optimum nutrient balance of iron for us that provides us with enough that we can live sort of relatively healthily, but not so much that encourages pathogens. And there is enough to support our immune system and our level of anemia that allows us to function, but not so much that you've got too much for pathogens. So here, this is pathogenicity, and this is the iron concentration. So somewhere here. So hence, supplements may favor the pathogen because you're pushing it over here. Whereas our negative acute phase reaction is there for a purpose. So during infection and inflammation, the body tries to sequester iron, and that has an important function, especially before the advent of modern drugs, etc., and treatment, and when we lived really exposed to so many pathogens. And another reason we think this is that when you look at the genomes of both people and the different pathogens, you can see that, particularly for pathogens, a large amount of the variation in their genomes is dedicated to acquisition of iron. So you can see that different pathogens, if you look at virulence, which ones are more virulent than the other is often related to how good they are at acquiring iron from the host. So a really classic example, which was sequenced by Brendan Wren at the London School, is Yersinia pestis, which is the cause of the plague. And there he showed that a huge proportion of the genome is related to different mechanisms of acquiring iron, and that pathogenicity is related to how well they acquire iron. And if you look at our genome, we also have lots of genomic variation, which is about controlling iron, and that may actually push us towards iron deficiency, but in the past may have been important in protecting us from disease. So this is a really nice little animal model that hopefully really shows this in action. So what you've got are these mice, just call them Balb-C mice. You've got two strains, let's get this right. So one is susceptible, so these red bars are mice that are naturally susceptible to this mycobacterial avium infection. And then there's these mice that are genetically somehow much more resistant to it. And so they took these mice and then they gave them different diets containing different amounts of iron. And then they look at how much the mycobacteria proliferates in the liver. And then there's another one where they actually look at how many mice die. So in the mice that are normally susceptible, then the amount of iron in the diet doesn't make that much difference to how many mycobacteria they find. But in the mice that are normally resistant, when you add more iron into their diet, that resistance disappears. Because that resistance, natural resistance, is about withholding iron. And if you then add in a lot of extra iron, you're stopping that resistance. And this amount of iron here is roughly equivalent to what we might provide in normal supplements. So it's not so much iron that it's totally unreasonable and not realistic. This is a realistic amount of iron that we might give people. So this is a really nice, sort of clear example of how this might work. And this is an even better one because this is in people. And this was done after I was not in the group anymore, but by my previous colleagues in the Gambia, in a very rural area. And what they did is they took adult men, healthy adult men, who are mildly anemic, and gave them a single iron supplement dose. I think it was 120 milligrams, which is what you would often give pregnant women as a daily dose, or iron deficient pregnant women. And then they took the blood 4 hours later. And then they took these, what we call sentinel pathogens, and looked at how well they grew in the blood. So the blue is blood before the iron supplement was given, and the red is the blood 4 hours after being given the iron. So here's your time 0, you add your pathogen, and then you're looking at the growth rate. So this is the growth in non-supplemented blood, and this is the growth in the blood of somebody who's just had a single iron tablet. So you can see how much faster the growth is occurring. And this is occurring for different pathogens here as well. So an E. coli, which is a common pathogen that might be making people sick. And then this is it's not Yersinia pestis, but Yersinia enterocolitica, which is also potentially important. And just to finish off with a nice example, Staph aureus, which we know acquires its iron through heme, has no effect because it gets it through heme. So giving an iron supplement, which is ferrous sulfate, is not going to make any difference. So to summarize the current best available summary of evidence and advice, the latest Cochrane systematic review on iron and malaria concludes that iron supplementation without screening for iron deficiency and anemia should only be done if effective malaria prevention or management services are effectively provided. But in practice, what does that mean? How do you decide that effective malaria prevention or management is in place? It's not entirely clear. And even when you look within the actual tables within these system, and there's been like 3 systematic reviews since that Lancet publication from Pemba, there's still evidence that it does potentially increase risk of clinical and subclinical malaria. So you can see more parasites after supplementation, even if not with symptoms. But overall, on a population basis, there may be more benefit than adverse risk. But how do you balance that population benefit to the risk of the few people that might get malaria? So a bit of summary. So the diagnosis of anemia. So to measure hemoglobin is relatively simple. What I didn't mention is that a HemoQ, many of you might have experienced with HemoQ, which is a simple point of care test, and it's a little thing you can carry around. It can run off batteries, and you do a finger prick, and it gives you an immediate hemoglobin, and it's accurate enough for surveys and things like that. But it's still quite expensive. You're still looking at least $1, and when I buy them in Japan, it's like double the price that I can buy it in Europe. But so you're looking at $1 to $2 per test, and you've got to buy the machine, which is about $3,000, $4,000. About $1,200 for the basic one. It's still too expensive though. Ideally, you want a test that costs 10 cents, 50 cents max. And of course, as we've seen, diagnosing the cause of anemia and iron status is still almost impossible in many cases. And then giving iron supplements blanket to everyone, it could be at best a kind of waste of resources, but there might be a real risk of increased infections. And also, there are many causes of anemia, and so only thinking about iron is not going to impact enough. We have to think about all the infections and the other nutrient causes. There was a nice trial, which I haven't shown, which shown a better impact on anemia from giving multiple micronutrients than iron alone or iron and zinc alone. I'll just summarise this, as I have mentioned Hepcidin already. So iron metabolism is complicated, and it's complicated for a reason, because we need to control it, and we need to make sure it's in the right place and not available for pathogens. But for quite a while, we didn't really understand how it was regulated. And then there was the discovery of this small peptide hormone called hepcidin. And then hepcidin seems to be the master regulator of iron status. So hepcidin is a negative control factor. So when hepcidin goes up, which it does during inflammation, or when you have sufficient iron, then what it does is it turns off the taps, as it were. So from the duodenum, from the intestine, it stops the absorption of iron, or in reality, it stops the transport of iron across the gut. So it turns off the tap here, and it also turns off the tap sort of here of the recirculating of iron, so that high hepcidin, you reduce your iron absorption and you reduce iron usage, and it's all sequestered. And then when hepcidin is low, which it would be in iron deficiency, then you're able to absorb and use the iron. So the signals for hepcidin is from the amount of iron stores, and then from the demand from the bone marrow, and also from inflammation. So inflammation pushes it up to stop iron absorption, and then low iron pushes it down so that you increase your absorption. So high hepcidin infection suppresses iron absorption, probably is a protective response. And then if anyone is particularly interested in this malaria story, there's now, although this is now a few years old, but new evidence of direct immune effects in controlling the liver stage of malaria. So again, nearly everything, we focus entirely on the blood stage of malaria, but first it has to go through a liver stage. And it might be at this stage that iron availability is important, more than the blood stage. And then the MRC Gambia group and a group in Oxford are continuing with what they're trying to look at is whether we could develop a simple point of care test for hepcidin that could determine who and when is it safe to give iron. Because if their hepcidin is high, there's no point in giving them iron because they won't absorb it. And then you're just dumping a whole load of iron into the gut, and there's evidence that then you're going to increase potential gut pathogens because now there's lots of iron floating around in the gut. So who to give it to and when is it safe to give it to them? However, it's still quite a long way from reality, unfortunately, and perhaps it's still not. Another level of complication might be there, and that regulation seems to be a bit more complicated than we thought. And then the other thing that might also help is these improved iron supplements. So the iron that we're giving now is mostly ferrous sulfate. When you talk to a chemist, we give ferrous sulfate, this big red tablet. Yeah, we give ferrous sulfate. A chemist will go, you do what? Because it's highly reactive, and you're going to have a Fenton reaction if you gave it with a large dose of vitamin C, you're going to get a nice Fenton pro-oxidant reaction going on. The absorption from it, you give this huge 120 milligrams, at best you absorb 10. What happens to the rest of it? So there's other formulations. So there's things like iron EDTA, which is perhaps a bit better, but not a whole lot better. So the next thing might be these nanoparticles, which is a colleague from Cambridge who's working on this, and that you might be then able to give much smaller amount of iron, and so that much more of it is absorbed and more safely absorbed. So hopefully that will help, and there are the first trials happening right now, I think. Okay, it's lunch. We can talk during lunch if anyone wants to come.